Hello, everyone. Welcome to the October session of the webinar series Topics at the Nexus of Climate Change, Fisheries, and Blue Foods. This webinar series highlights the impact of climate change on fisheries, aquaculture, and the communities that depend on them. You'll see on your screen the logos of the Ocean Decade programs and projects that are behind this webinar series, and I'll introduce them really briefly. Blue Food Futures Program, Fisheries Strategies for Changing Oceans and Resilient Ecosystems, or FishSCORE, Sustainability, Predictability, and Resilience of Marine Ecosystems, or the SUPREME Program, Sustainability of Marine Ecosystems through Global Knowledge Networks, SmartNet, and Fisheries and Marine Ecosystem Model Intercomparison Project, FishMIP, as well as the endorsed project, Basin Scale Events to Coastal Impacts, or BECI. We're very excited to have the topic of today's webinar be ecosystem modeling to inform resilient fisheries. And I'll kick it off to our first presenter and introduce Stephen. If you have any questions during Stephen's presentation, feel free to drop them in the Q&A box and can address them after the presentation. But I'll introduce Stephen. Stephen Guidos is a PhD student at the Norwegian Institute for Nature Research and the University of Bergen in Norway. His main focus is on the cascading impacts of fishing pressure within nearshore marine communities in Western Norway. Stephen also aims to determine possible impacts of fisheries on terrestrial systems by studying dietary change in semi-aquatic predators related to fishing pressure. Stephen, thanks so much for sharing your work with us today.
Yeah, thanks for having me, guys. I really appreciate that. And thanks for the introduction, Laura. Yeah, as Laura said, my name is Stephen Guidos, and I'm a PhD student at the Norwegian Institute for Nature Research and the University of Bergen. And today I'm going to be talking about some of the recent progress we've made on mapping the commercial wrasse fishery in Norway. Yeah, so if we can go on to the next slide. Yeah, so the wrasse fishery exists not for consumption. It exists entirely to support the salmon aquaculture industry in Norway. And so, yeah, there's no consumption related to these fish at all. So they're captured live. And the point of using wrasses is to use them as a parasite control. And so they're captured live from Norwegian ecosystems, and then they're transported to salmon aquaculture farms, where they're essentially dumped into the farms together with the salmon. And the idea is that they will eat parasites off of them. And there are 5 species of wrasses in Norway. But if we go to the next slide, you can see there's really only 3 that are mainly focused on, and that is the ballan wrasse, the corkwing, and the gold cinny wrasse. So we'll be mainly focusing around those 3 as we move forward here onto the next slide. So just to give you a bit of a background on the Norwegian salmon aquaculture industry, it's the second most economically valuable industry in all of Norway, just behind oil. So the government has a vested interest in making things go smoothly. However, it's been facing some challenges, and the most destructive challenge, or the biggest challenge, I guess you could say, is related to parasite outbreaks, and specifically salmon lice. And so if you go to the next slide, you can see what salmon lice actually look like. They parasitize the outside of the salmon. Basically, they eat the gills or any soft material, including the eyes, the skin, the mucous membrane, which is a huge problem for the fishery because it results in reduced growth rates of the salmon, and you can't market the salmon for the same price if they have salmon lice on them. And so eventually the salmon lice will kill them if the outbreak is bad enough. And so it's a huge economic loss to salmon aquaculture, but more than that, it's a really big ethical concern because we're losing millions of fish in a kind of tragic way. And there's several ways to treat lice parasitism, and one of the first was chemical treatments, where you basically submerge the salmon together with the lice inside of a chemical bath. And then this would hopefully remove the parasites, but these salmon lice reproduce quickly, and now they are resistant to chemical treatment, so that doesn't work so well anymore. They also use hot water and pressurized water to remove the salmon lice, but as you can imagine, that's very stressful for the salmon as well, and many die during that as well. Again, a huge economic and ethical concern for the salmon aquaculture. And so that's where wrasses come in. Wrasses have been kind of touted as the environmentally friendly method to combat salmon lice. And so if we go to the next slide here, you can see that wrasses are essentially captured from the wild, and then they're put in the pens with the salmon. And the idea is that they swim around the salmon, and when they see these salmon lice on them, they swim up and they eat the parasite off of the fish. And so that's the artist's depiction of what it looks like. But if you go to the next slide, you can see what it actually looks like. And so wrasses, for the most part, are, I wouldn't say they're demersal fishes, but they're highly associated with benthic habitats. They live mostly around kind of boulder areas and rock bottoms, or around macroalgal growth. And so you can imagine that being put in a salmon pen, which is an open water environment surrounded by tens of thousands of predators, is not, well, it's quite a stressful environment for them. And what's worse is that the wrasse cannot survive overwintering in the salmon pens. So there is close to a 100% die-off of wrasses each year. And that means that they need to catch new wrasses from Norwegian ecosystems every year to support the fishery. And as of now, there's a quota of 18 million fish that are captured per year. They haven't quite met this quota in the last couple of years, so the fishery might be in a bit of a decline at the moment. But this really begs the question, what does this do to the wrasse populations themselves, and what are the larger ecological ramifications of the fishery? The wrasses are an important dietary component for other fish species like cod, and other animals eat them, like seabirds, semi-aquatic mustelids, and wrasses themselves are important predators for things like mussels and clams. And so, yeah, and one of the nice things about wrasses and about modeling them is that they spend their whole lives in a very small space in the same stretch of coastline. And so if we can model fishing pressure on a very specific scale, this should only relate to the fish within that small area. They don't migrate. And so repeated fishing in an area should have local impacts, which will make available kind of ecological studies later. So if you go to the next slide, though, you can see the only problem is we don't really know where they're fishing. At least we haven't historically. And so the only data we've had on catch statistics on the national level is on the municipal level, and that's just through sales slips. So all we can see is how many fish of each species are sold in each municipality, but that's not very specific. If we want to go and see what's happening to the wrasses, we need a way of actually determining how many wrasses are caught from each little piece of the coastline, and go to those areas and do some surveys. And but luckily for us, if we go to the next slide, in 2020, the Norwegian Directorate of Fisheries recognized the need to figure out the extent of this fishery. And so since 2020, every single vessel in Norway must be equipped with a GPS device of some sort, which will track the boat's movement throughout the entirety of the fishing season. This is, it's like a blanket rule for everyone in the fishery, aside from the youth fishermen. But that's a very, very small part of the fishery. We're talking like 1 to 2% of the total catch. So it's okay that we don't have that data. But since 2020, every vessel will have a date of sale for every time they sell fish, how many of each species, and it will also include spatial data in the form of AIS or VMS data. And if we go to the next slide, we can see that although it's helpful to have this data, we can't really tell exactly when the boats are fishing based off of their spatial data. This needs to be inferred from the speed at which the boats are moving. So the vessels do not need to keep a log of when they start and stop fishing. So we need to infer this from the spatial data itself. And so we went out and collected a key data set. We can see on the left-hand side, the red lines are when the boats are traveling. Green is when they're pulling up the pots, and blue is when they're setting pots. Yellow is when they are delivering to the salmon farms. And we can see that there's a different speed value set kind of assigned to each of these different activities. So if we click one more time on the slide, we have developed a kind of reference data set. So in general, the boats slow down when they're deploying and hauling the gear, and they speed up when they're traveling. And there's a very nice cutoff rate here of about 4.2 knots. Over that, they're traveling, and under that, they're probably fishing. In addition, because the coastline of Norway is so steep and the wrasses are associated with the bottom habitat, they're like coastal, like rocky formations, in the littoral zone, all fishing pretty much happens very close to land as well. So that's kind of an extra parameter that we can use to filter out the traveling data, because we're talking about a data set of many, many millions of points. And so the more we can get rid of, the better. Yeah, and technically the optimal distance to land is about 50 meters, but we increased this to 100 just to make absolutely sure we're not cutting out any data we don't need to. And then if we go to the next slide, we can see once we have this data, we can actually start modeling the fishery. The fishery is modeled in 9 steps. So we have a data import, and then we do a data quality check, and then we start filtering the spatial data. We get rid of the data over that speed threshold and outside of the distance parameter. We get rid of all the data inside of ports and within the salmon aquaculture pens and a few other things. But after we're done filtering the spatial data, we actually need to break the data into 2 separate data sets. We get an AIS data set and a VMS data set. And we need to do this because the AIS data and VMS data are, on paper, they look the same, but they're collected at a very different rate. VMS data is collected once every 10 minutes in this fishery, regardless of what the boat is doing. If it's at port, if it's at fishing, it doesn't matter. It's 1 point every 10 minutes. That's not the case for AIS data. So when a boat is traveling quickly, it will plot many more points. And when it's slowed down or at port, it will plot fewer points. And so you can't directly compare the data to each other. We need to treat the AIS data as tracks. So we connect the data points. We don't do this for VMS data because individual fishing like bouts usually last 10 minutes or less. And so there's no point connecting 2 points that are when the ping rate is 10 minutes. So you'll basically be connecting different areas or different fishing fields, which isn't good. So after we create the AIS tracks, we combine the catch statistics with the spatial data based on shared date. One of the nice things about this fishery is they catch fish and sell it on the same day. So that's very easy. And then we combine the catch data and the spatial data with these polygon matrices in which we can summarize the fishing effort in different parts of the country based on shared geometries. And at that point, we have an AIS data set and a VMS data set, or an AIS and a VMS map for each year of these different polygon sizes. And the last step is essentially to combine the two based on shared polygon ID. And you can see on the right-hand side here, we have mapped 2021 through 2023 at 3 different scales. We have the salmon production area scale, we have a 5-kilometer grid, and a 1-kilometer grid at the very bottom. And I know it's hard to see the polygon grid sizes in this format. So if we go to the next slide, you can see there's a bit more of a difference in the 5-kilometer grid and 1-kilometer grid. And all of the maps are completely interactive. So if we go to the next slide as well, and click the play button, we can zoom into any region of the coastline that we want to. This is the 1-kilometer grid in 2022. And so we can also toggle on and off the polygons, and we can see where the salmon aquaculture pens are and the different boat harbors. And then we can zoom into a specific region we might be interested in. And if we click on a polygon, you can see that the total catch that year was 15,000 fish approximately. there was a coastline length of 5.4 kilometers, which gives us a catch per kilometer of about 2,800. And it's split by the different species as well. So you can see which species are most targeted. In this case, I know the species are listed in Norwegian. I'm sorry about that. Grønnhjelt means corkwing wrasse. So corkwing wrasse is by far the most heavily fished species in the region. And you can go to any region you want in the country and you can basically see what the fishing intensity is like. Yeah, and then we can go to the next slide. And once we have this data and these different maps, we can essentially compare catch per unit effort between polygons over different years. And so the x-axis of this, all the plots on the right is essentially catch per unit effort in year 1, and catch per unit effort in year 2 is on the y-axis. And as you'd expect, it increases linearly up to an extent. So if you have a low catch per unit effort in year 1, you can expect the same catch per unit effort in year 2, right? You can sustain low fishing pressure for a long time. But if you exceed a certain threshold for every species, after a certain point, you will start to see reduced catches in year 2 following high catch rates in year 1. And this was the same for ballan wrasse, goldsinny, and corkwing. Makes sense. Those are the most heavily targeted species. Rock kook did not show this. Rock kook was included just as a bonus here, essentially. But catch statistics vary wildly between the years. It's not every year they catch a lot of rock kook, and it's quite randomly distributed throughout the coastline. And so that population does not seem to be in danger of being overfished at the moment. But one of the interesting things was that the regression lines were statistically much more significant at the 1 kilometer grid level compared to the 5 kilometer grid, which kind of indicates that localized resource management of these wrasse fisheries is much better done at a smaller local level. And that makes sense because the 1 kilometer grid will include fewer local populations of wrasses. The 5 kilometer grid could include many different wrasse local populations, so you could pretty much alternate fishing pressure between those in different years. But on the 1 kilometer grid, you're probably targeting the same exact wrasse populations every year, year after year. So it makes sense that the strength of the relationship is different there. And then on the next slide, there's one more problem with the, at least one more problem with the salmon aquaculture, and it's that they have recently had some disease outbreaks of infectious salmon anemia, which means that in these ILA zones, they're called in Norwegian, you're not allowed to transport any fish into or out of the zones. You're allowed to catch wrasses in the same zone as you, if you have a salmon farm in an ILA zone, it can technically have wrasses, but the wrasses must be caught in that same zone, which is inconvenient for many fishermen because it would mean they'd have to switch up where they're harvesting the wrasses. And we wanted to figure out, does the establishment of these zones have any impact on the catch rates of different wrasse species? And so if we go to the next slide, we can look at the catch rates. So we have a control region that was essentially the rest of this municipality, and compared to the ILA region. And you can see from 2021 to 2022, the catch rates decreased substantially, kind of universally in that area. But after the ILA region was established, we see again a significant drop in catches in that area compared to the control region, where there was no difference in catch rates between 2022 and 2023, which probably indicates that the wrasses are experiencing some level of protection from fishing pressure after we implement these kind of not no take zones, but no transportation zones, I guess you could say. Which is interesting. And if we go to the next slide, now we can talk about what we can actually do with these models, right? Now that we know where they're fishing, we can go to these areas and actually see what's happening to the wrasse populations. That's not exactly my job in the project. That's another student's job. But I thought, hey, I do a lot of work with otters and mink. And I thought, why don't we use these guys to help us and figure out if there's any kind of change happening to these fish populations based on how many wrasses these guys are eating. And so wrasses are a huge dietary component of both of these species. So any changes to the wrasse populations should be reflected in their diets and larger ecological ramifications if we get enough data. And so last year I went to this marine protected area. That's this red polygon. And we collected about 150 otter and mink scats from there. And these will be compared to about 150 otter and mink scats from these fished regions. And so we have two methods of doing these analyses. If we go to the next slide, we can look at the actual physical remains in the otter scats, and we can look at vertebral structures and otoliths to actually determine the age of the fish that they're eating. You know, you would expect in a marine protected area, there's no fishing for wrasses, so we'd have larger wrasses, older wrasses that are being eaten compared to fished areas, but maybe even higher biodiversity. I'm not really sure how the marine protected area might affect other fish species as well. And if we go to the next slide, we can also run some meta barcoding on the diets. And so this was something I was busy with last year, and I'm still crunching through the data at the moment. I've been busy with some other papers. But we collected some preliminary data back in 2022 just to make sure that this was actually going to work and be a viable method. And we just took a trip to the MPA and another fished region. And you can see those kind of red and green respective polygons. And the fish we care about here are the orange and the yellowish kind of bar plots there. Those are the wrasses. And we can see, you know, again, we have to take this with a huge grain of salt here because this is a very small sample size compared to what we just collected. But we can see that the otters were eating more wrasses, or a larger number of otters that we had data from were eating wrasses compared to the fished area, and that there was a higher dietary component. So yeah, that's yeah, again, take it with a big grain of salt. That's a small data set. So maybe I can come back in a couple of months or a year and present the full data set if anyone's interested. But yeah, that's pretty much all I had to say today. So if anybody has questions, please just let me know that.
Thank you so much, Stephen. If anyone has questions, feel free to put them in the Q&A box and we can read them. We probably have time for one, maybe two questions before we turn to our second presentation. Hearing none at the moment. I will introduce our second presenter. If anyone has questions for Stephen related to his presentation, feel free. Oh, there's one that popped in, so I will ask that and then we can turn to our second. We have a question from Rich Bell. How effective are wrasses at controlling salmon lice?
Yeah, that's a great question. Yeah, we think one of the reasons why the fishery has been decreasing is because, you know, there is this whole ethical perspective to it. You know, is this actually the right thing? But we also believe that some of the salmon farms don't believe that it's actually an effective control mechanism. One of the problems is with some of the species, especially the gold cine, they're quite small. And so they escape through the holes of the salmon net quite fast. That's why most people want ballan wrasses. Ballan wrasses are the biggest, but they're also the least numerous. So that's a big problem. So that's why we have very strict size limitations on harvesting ballan wrasses, actually. But good question. The answer is we don't really know for a fact how effective it is, but considering the fishery is slightly decreasing, probably not super effective.
Thank you for that. There's one other question then in the chat at the moment from Jay Kim, who asks, how does the wrasse fishery compare in scale to lumpfish aquaculture for sea lice control?
I believe the wrasse fishery is still a bit bigger. One of the advantages of lumpfish, though, is that they're a bit wider, so they don't escape as much, and they can overwinter a bit better. So I think some farms, especially up north, rely a bit more on lumpfish. I don't have the exact numbers in my head, though, as to how many lumpfish are being used in salmon aquaculture. And there's also, I think, a difference in because they're also using aquaculture to raise lumpfish and trying that with wrasses as well. I believe it's been slightly more successful with lumpfish. I think they're a bit easier to raise in captivity, but I will have to double check on that. I'm sorry, I don't have a good statistic in my head at the moment.
Oh, good. We can also share, as part of the recording and things like that, we can share your contact information if you're comfortable with that, so you can follow up afterwards.
Yeah, please do. Just send me an email if anyone has questions.
Perfect. Well, thank you again so much, Stephen. If anyone has more questions for Stephen, feel free to put them in the Q&A box, and we can either answer over text or when we get to the end of the webinar portion. I want to turn it to our second presenter, Dr. Carlos Robinson, who is a biologist who graduated from the Faculty of Sciences at UNAM and holds a PhD from Bangor University in the United Kingdom. Since 1994, he has been a researcher at the Institute of Marine Sciences and Limnology, where he currently leads the Fisheries Ecology Laboratory. And Carlos will be presenting on a decade of warming and declining productivity in the Gulf of California, exploring the drivers. Carlos, the floor is yours.
Thank you very much. Thank you all for the presentation. So I will share my...
Oh, Carlos, I'm sharing the screen so people can see it from my screen. So just tell me when you want to do next slide.
Can I use my PowerPoint?
Unfortunately, we're not able to do it that way. So just
So-
if people can see your slides, so just let me know when to advance your slides.
Okay, so okay, all right. So well, this is the part of the research we are doing in the Gulf of California. Is the next topic, please. The next slide. So the overview is the Gulf of California is a semi-enclosed sea with exceptional diversity and zooplankton standing stock that actually surpasses many comparable marine environments in the area. No, the Gulf of California contributes more than 70% of the total catch. At the time, it contains actually more than 10 marine protected areas, making it the most protected marine regions in the country, in Mexico. So over the past decade, notable changes have been reported in marine species, fisheries, and the epipelagic ecosystem. So this region primary productivity is largely influenced by wind-driven upwelling system events because of the wind. Therefore, it's essential to focus on variables affecting wind strength and direction. And that's the idea of the results of this investigation. Next, please. So this is the Gulf of California. This is a semi-enclosed sea. In the north is United States, and the west is the Baja Peninsula, and the east is the mainland. So it's about 150 kilometers long, with many islands and many protected areas, as I said. Yes. The important thing is that most of the productivity in the area is produced by winds and by the tides in the upper part of the Gulf of California in the main inland region. No? So this is the study area. And it's a study area interesting for using satellite data because as you can see, and actually it's really, I say that it's an area with very low cloud coverage. So it's to use satellite data is useful because of the clearness of the area. No? Next, please. So what I am presenting here are some results using two sensors. One is MODIS for sea surface temperature and chlorophyll, and the other is the MERAS for wind speed and air temperature. Air temperature actually is at 10 meters high. So these are the two sensors that we are using. And as you can see, in blue is the MODIS 4 kilometers area. And in red is the area of the MERAS for wind for wind speed and air temperature in the area. So all the results I'm going to present in the next slide is this area, which is actually the central south part of the Gulf. Is not included in the north because in the north is more of tidal mixture that is not related with sensor data, satellite data. So this is the area. Next, please. So we have the results. So then, interesting, this is the monthly data anomalies for 2025. So it showed the wind speed, the sea surface temperature, the air temperature, and the chlorophyll data. These are monthly data, these are anomalies. And one interesting thing is that you have this continuous line in the fourth graphs. And actually, this is the partial sum of the anomalies. No, this is a rapid methodology to see tendencies in the data. And for the 4 events, wind, sea surface temperature, air temperature, and chlorophyll, when you use these parsumer anomalies, there is a clear pattern divided in 2 parts. No? This before 2014 and after 2014, as you can see. If we see the wind speed, you see the area is continuous with the high anomalies, positive anomalies of wind speed until this date in about 2013-14, and then decline. And corresponding the sea surface temperature, the temperature passes from an environment of cold temperature before 2014, and then changing quickly to an environment of warm temperature, as observed by the tendency of the parsumer anomalies, and you can see the data. No? So the next is the air temperature that follows a similar pattern at the sea surface temperature, as expected, because sea surface temperature and air temperature is more or less the same environment, no? The 10 meters high and the sea surface temperature. And again, before 2014, there was an air temperature environment of cold environment, with most of the anomalies are cold anomalies in the area, these mountain anomalies. And then a sudden change in 2014, as you can see in this part. You can see the positive anomalies and the change of the pattern in the area. No? And then you see the chlorophyll data come from high productivity area with all these high positive anomalies, to going down in this part in 2012, and then going down from 2013 in this case, and so on. So in fact, we have two different session of times. No, is one from 2000 to 2014, and then the other. No, there was a change of wind intensity, was a change of warm temperature, cold temperature to warm temperature in the area, the same in sea surface temperature and air temperature, and it was a time that this from 2000 to 2014, yes, with high chlorophyll, and then sudden decrease as well. Following, and the interesting thing is that the 4 variables change basically in the same time and in following the same pattern. So the next question is, what is the effect in the ecosystem of the Gulf of California? What is the effect of the fisheries in the Gulf of California? So this is the thing that we're going to see. Next, please. So, well, this was the line observed. So this is the observed changes in behavior of fisheries and marine mammals and the pelagic ecosystem. Next, please. So it's interesting, no? This is the squid, the jumbo squid, Dosidicus gigas, and you see this how the squid size changing from 2000. This is a photo both photos I took. This was in June 2014, and then in 2016, we did some research vessel there, and we found the change of the size of the squid. No, both are mature individuals. So we are not in 2016. We were not catching the babies. No, they are mature individuals. This is a change in behavior and form and phenology of this species, which is very common even in the southern part in Peru. That this animal change size according to productivity. Next, please. Then we have the jar sardines and squid catches, and you see the line 2013. And before 2006, there was a lot of catch of sardines, and then going down after 2014. The same happened with the squid catches. Very high catches before and then after the area. So it was following more or less exactly the same pattern as with the previous environmental variables. Next, please. So apart of species like they are fisherable species like sardines or squid, so we have some observation with blue whales observation with Tometal in 2023, and you can see there were high numbers of well observation in 2012 and going down, and as expected by the changes in the environment and the variables I show you. So the reduction of well observation is evident in the Gulf of California. Next, please. And then we did also oceanographic surveys during this time. We were doing the oceanographic surveys before and after all these events, and before 2014 and after. So next. And one of the things is consideration. We use ecosonders to part of the reservoirs using ecosonders to evaluate myctophytes and the deep scattering layers. And this is just a way of observing ecograms. No? It's just intensity of eco integration. And you see in July 2007, there was a lot of configuration. So, and then the configuration is going down. It's got in blue, all the area, getting down to blue. And some of we saw another big scatters down in 2019, but nothing compared with the we observed in 2007. And we have the numbers, but I just want to show you here how the ecograms look, and you can see the different pattern. No? A lot of configuration in these years and going to almost very few in 2016 and so on. No? This is one of the parts that we observe along the water column. Next, please. So then we did some results using ZTDs and yeah, of course, the first graph is the normal behavior of the temperature along the water column, which is hot, warm during the summer. As you can see in the first figure, this is the normal pattern, no? But if you see along 2016 to 2010, as you can see also an increase on warm temperature along the water column. That means that the thermocline decline is a big water column with warm temperature in 2015. So similar as we saw more or less using satellite data. Next, please. And then we did also sampled with zooplankton sampling, no? The standard sampling of bongo during these cruises. We use these bongo nets, as you can see in this graph. The idea was to sample basically krill, faucets, yes, in the area. And next, please. Yes, interesting is this graph is because we have seen, we analyze all the data from the bongo net, and interesting that we don't see any change of the mean abundance of the faucets principally, no? So that means that for each cruise that we did, we analyzed the abundance of the faucets using the standard methods. We don't find any difference on the abundance, no? But if we look for the size of the swarms, of the swarm, yes, and with this analysis using the cumulative summatory of the data, results showed, and it's interesting that in fact, in this is 2005 2014, the black line, and then 2015 to 2019, the red one. No, the analysis showed that actually the big swarms of fascidos that was reduced before 2014 was you find big swarms, no, comparing with all the size that we found, and And recently, and this was until 2019, we have more data until 2013, but after that, you remember it was the COVID, no? But results show that interesting, the swarms of fossils would just get smaller. There was not big swarms. Next, please. Okay, so understanding recent change requires a long-term perspective. Yeah, that means going back to 1980 to 2005, 2025. Next, please. So we have for Gulf of California, we have two different etapas. I am analyzing wind speed, temperature, and with El Niño and PDO, and we can find three different eras or times in the time, and I am using the change of wind speed which occurred from in 1998 and then again in 2014 when the wind declined, and here was the increase of the wind. So we have three different eras of years. One with moderate wind speed, lower to mid temperature, with several niños above 1 in this time, as you can see, all these niños, few niñas, and the PDO basically positive around the time. So the next time was the productive area with the spin very high, low temperature, few niños, and the PDO is going down. And the next part, which is the part that we are analyzing down, this 2014, 2025, wind speed is starting to increase, but no without the force that we had before, no? And then the apparent temperature was evident, the change of temperature in the area, this is what you see, and we are here now. And in this time, there were two El Niños, to 2013, 2023, and the decline of the PDO, as you can see here. Next, please. Then, to understand what happened with the wind, we need to study the pressure system in the area. Basically, it's dominated by high pressure in the Pacific, high in the ocean, and the low pressure in the land. No, so this is the two systems of high low pressure in the area. Next, please. So we use I use all these points of Meras. And we have the red dot where in the wind anomalies in the Gulf of California during the winter are calculated. The idea is what is controlling the wind speed in the Gulf of California. Next. So we have this is only because of time. I'm only presenting results for March, and you can see that the area was controlled. The wind speed in the Gulf of California is this area of high pressure, exactly here in the Pacific, no? And interesting that we only analyze this area here in this area. That means the pressure and temperature are inverse. No? The higher temperature, the lower the pressure, and therefore the lower the wind intensity. Next. And then we have all the analyzing only the Pacific anomalies, the area with high production, with the high correlation with the wind in the Gulf of California. Again, we have the three different eras with air temperature in the more or less average, and then drop the air temperature in the area of high pressure that control the wind in the Gulf of California, and again. And then this change of temperature very high in 2014 and going down now here. The air pressure, therefore, was average during this time, high between 1998 and 2014, and then the air pressure is increasing, but not as strong as before. And then we have El Niño and all these variables that you can see as before. Next, please. So interesting is that we analyze the area that controls, let's say, the wind speed in the Gulf of California, and we correlate the air temperature with the PL, with both indices, with the Pacific Decadal Oscillation and El Nino, and the temperature is more correlated with el PDO, as you can see in this graph, no? But if you analyze, if you see the residuals of the analysis, interesting that during the last, from 2014 until now, to this year, the residuals of the regression are positive all the time. That means that the area is getting more warmer than as expected with the PDO, and there are no more relationships, not as strong as with El Nino. Next, please. So if we analyze, I assume these Pacific winter anomalies and certain catches in the Gulf of California, interesting is that the most of the capture of sardines in the Gulf of California were presented during when in the Pacific area where the area where controlled the wind speed in the Gulf of California was with a low air temperature and high pressure, and then you have the sardine catches maximum during this time. Next. So interesting, and this is almost the final, is from all the cells that we analyze in the area, something that is very interesting that from January 2014, almost all the cells of temperature were positive, as you can see in this graph, This was the time with the low temperature and then change for increased temperature. But what take my attention is interesting that the whole area was warm, and when I say the warm area, all these particular cell, That means that this is a change of temperature, at least from 1980 to the present, we haven't seen before this change. It was a warming, very intense warming, and continuous. There was some reduction during this time, but it's obvious that the change of temperature was huge and was wide. Next, please. So basically, these are the conclusion. During the last decade, change in wind pattern played a central role in driving ocean warming and biological changes in the Gulf of California. The onset was in January 2014, but the analysis showed that it was not directly with the PDO or El Nino, because during this time, in January 2014, there was the Nino, El PDO was almost negative of 0. The center of the near surface air pressure associated with the wind speed during the winter in the Gulf of California is located off the Baja Peninsula in the Pacific Ocean. And there are different surfaces where near surface air temperature temporarily influences pressure. That means that the pressure that controls the wind is regulated by temperature. That's important, as this is the thing that we have to to look for the change in the temperature in the area. What distinguishes the recent period is not only the magnitude of the warming, but also this exceptional duration and special coverage. As I was saying, this is something not seen before. With this, I think I finish. Next, please. Yes. Thank you very much for your attention. I appreciate and thank you, the organizer, for inviting me. This is the work we have done we are doing in the Gulf of California. Thank you very much.
Thank you very much, Carlos, for your presentation. If anyone has questions for Carlos or Stephen, feel free to put them in the Q&A box. I guess I'll ask to kick us off. I'm curious if you could talk about some of those like locally focused studies that you've been doing recently and what those look like compared to some of the previous work.
Is that for Carlos, Laura?
Yeah, for Carlos.
Okay,
So-
yeah, okay. Yeah.
Sorry, again, please.
You mentioned that some of the ongoing work is focused on local conditions and how those might be sort of impacting warming and productivity. And I'm curious sort of what the current research looks like compared to this sort of long scale study.
Well, part of this program that we have with the decade is that to understand and to search for local responses of anything from plankton, from whale, from fisheries, from different parts of the area. So what we are trying to find out is from all these different locations, if they've noticed, because there are local fishermen that they work there and they do their job. We are trying to do, and now we are doing, is if there is any change. I mean, the question is, we say, have you seen any difference between the catches now and before? And this is the research we are doing now to go to the locals areas and for the local people with the fishermen, yes. And yes, there are some fisheries that they're not very important, that the sardine of anchovies or squid. No, they're small fisheries, but you see the result and you see the graphs all follow the same pattern as we see with the big fisheries. No?
Well, thank you. There are two questions in the chat. I think both are for you, Carlos. The first is from John. Were there any impacts on phytoplankton species composition or dissolved oxygen near the thermocline? And the other from Rich, are there species that have increased since 2014?
Okay, talking about regarding species composition of phytoplankton, we don't have the data, I'm afraid. No, we have basically because most of our research that we did and we do, I mean, in the oceanographic cruises, is to analyze plankton, and we use I mean, all this phytoplankton analysis, but not for the species. I don't know actually. And the second, sorry.
The second question was, have there been any species that have increased since 2014?
Yes, if you see the graphs of the catches of these pelagic species that are very important inside the Gulf of California, it's incredible that the anchovy species, the anchovy, the northern anchovy, yes, the captures are, I mean, the opposites of the sardines, going up and the decline of the sardines. No, that means that yes, for an environment, a warm environment with probably another species of phytoplankton, something, but with low productivity, species like anchovy flourish. They are more abundant, and well, actually, it's something like a behavioral fisheries, because if you don't find sardines, that usually is the more productive and means more valuable species for the fisherman, for the industry, the Monterey sardine, they try to catch that. If they don't find this, then they change to anchovy. So, in fact, what we are seeing increase is the capture of sardines, of anchovies, but not really if the abundance of anchovy is increasing. Because we only have data from fishermen, you know? So there is low sardine, then they go for anchovies. Then the anchovy case. It not necessarily means there are more anchovies, but maybe, we don't know. But a good question is something that we have to look after, no?
Makes sense. I recognize we're getting to the top of the hour, and wanted to, unless there's a last question that comes in, wanted to thank both Stephen and Carlos for your presentations today and for joining us, and to thank everyone that joined for participating in the webinar. A recording of the session will be available on the GMRI YouTube channel with a summary of the session, and we'll be sharing sort of contact information so that you can reach out with any additional questions. I don't believe we've decided on a date and time for the November webinar, but keep an eye out for an email coming out about the next month's presentations. But until then, hope you guys have a